It's one of the biggest logic errors that's made in science, and whenever it comes up, it's a real big one you got to pay attention to. So they were able to see bacteria in some diseases and said that that was the explanation of the cause of those diseases, but there were others that remained unable to find an organism. And poliomyelitis was one of those. So what they did was they theorized that there was actually a poison, a toxin, that came from somewhere, and this is what a virus was. And that's why the Latin root of virus means poison. And they thought that this was a transmissible type of poison that could actually reproduce in your body, though. And this is what makes it a very unique and special type of poison that deserves a special name of virus. Okay, and so what they would do is they would have a sick animal, and they would filter out the diseased part of their tissue, like I described in that purification experiment, to remove cells, cellular debris, but it doesn't remove everything. And then they would take that filtered material, okay, and they could make other cells sick with it, okay? So this was what they thought a virus was. However, they were really not able to see it, ever. And when the electron microscope came out around 1930, they really were hopeful, but it never panned out. They could never see this kind of protein poison virus that they were theorizing. Okay, so essentially the field was not very successful, and it was sort of collapsing in on itself. Okay, but then a couple of things happened to revive it. Okay, and this man, John Enders, is a very key figure in this revival that led to this modern dogma of virology that I described with the current situation. So one factor was, in the early 1950s, the discovery of double helix of DNA was made public. And this was like a new beginning in biology, that the importance of this genetic molecule was going to transform our understanding of everything, and including viruses. So this is a very key occurrence. And the second factor has to do with what was going on with vaccines. And Enders was very successful in, he is actually the one that came up with the scientific idea behind Salk's poliovirus. But then he kind of bowed out and let other scientists develop the idea and get the credit for it, but...
Hi, this is Chris Olson from Treasure Island Coins coming to you.
Just a brief mention from our sponsor. So anyway, the Salk polio vaccine did not go well, and Enders was pretty critical, and he decided that he wanted to focus on measles going forward. So at the time, the virologists who were studying this, they thought that taking that filtered material that they called a virus, that it would only affect the same exact type of tissue that the person who was sick had. So for example, if it caused pneumonia, then it would only affect lung tissue, and that it wouldn't cause any damage to other types of tissue. But Enders had this idea that maybe it could affect other tissues, and maybe, and with the polio vaccine, it was actually used, it was cultured in fetal tissue. And then he had this idea that maybe it could be in other animal cell cultures. So this study in 1954 was critical, and it basically laid out the modern experiment to prove a virus. But it does something more than that I'll get to in a few minutes. So what I discussed in the beginning about mixing the rotting flesh with starving, poisoned cell culture, that comes from this paper. And what he did was he found some kids with measles, and he took nasal swabs and washings and such, and put that in some various cultures, and he used monkey kidney cells. Okay, this is the first time these monkey kidney cells, and if any of you have heard my lectures, I mentioned monkey kidney cells quite a bit, because they're the favorite cells used in these experiments. And so he was able to, what they call this, propagate, because he would put the infected tissue in with one culture, let it incubate until it showed damage, then take liquid from that, filter it, and put it in a second one, and that showed damage as well. Okay? And so that was the main accomplishment. And I brought this paper because I didn't have it at the time I did my slides, but I wanted to read what he said in his conclusion section about this, okay? Because he changed his tune after this. He wrote, while there is no ground for concluding that the factors in vivo, that means in a living organism, are the same as those which underlie the formation of giant cells and nuclear disturbances in vitro, okay? So there's no ground for concluding that those factors is consistent with the properties that a priori might be associated with the virus of measles. Okay, let me translate that. They saw damage in the cells in the laboratory culture. He's saying there's no reason to think that's the same thing that what actually happens in the person with measles. Okay, so in other words, he's saying this experiment does not prove causation. He said there's no reason to conclude that it proves causation. Okay? So he was very critical of these techniques, and it was mostly a way to manufacture vaccines at the time. But what happened is he was awarded the Nobel Prize. He had lots of companies coming to him, giving him contracts, wanting his advice, etc. And he changed his tune about this line of critical analysis of his science and started to say that it was a cause. And this is really what I call the master virus dogma strategy here. So you take this recipe on the left, where you have rotting flesh from a sick person, mix it with poison in a starving cell culture from monkeys, and not only do you get the cytopathic effects that are quote-unquote proof of a virus without actually showing a virus, but also this is the recipe to make a vaccine. So that experiment I described before with the mice, what they essentially did was they put a vaccine in those mice without the adjuvants. Okay, because you take this cell culture product and add adjuvants, that's a live virus vaccine. Okay, if you take it out of that culture, put it in another culture, let that grow, take the fluid out, put it in another culture, and then put it in with the adjuvants, then that's an attenuated virus vaccine. Okay, so this strategy, which doesn't prove anything, according to the scientist himself, is now taken subsequently for every pathologic virus, right? Every virus that causes disease, this exact technique is what is given as proof of the virus, and this exact technique is what's used to make a vaccine against that virus. And this is the deepest fraud behind everything else I mentioned that's been perpetrated upon us. So to summarize, we have many, many levels of fraud in this pandemic situation, in isolating viruses, in the diagnostic test, in showing causation of an illness, in sequencing the genome, and of the underlying master strategy, right? Where we can prove a virus and essentially make a new vaccine at the same time. Thank you everyone for listening.